Hello, my name is Deidre Beavers, and I am a software developer on the Reactome project. Today, I will be discussing the Reactome IDG project, which aims to place dark or understudied proteins and place them into the context of reactome pathways. Reactome is a large and highly annotated pathway knowledge base, so it is great for creating a context for dark proteins to predict their function and potential therapeutics. The Reactome IDG project uses a random forest model that was trained with 106 different pairwise relationships from a variety of sources. The functional interactions from Reactome were used as a training set. Together, this performed very well with an area under the curve score of 0.89. The Reactome IDG web portal has several useful applications. For example, the user can enter in a certain protein and see the pathways that are predicted to functionally interact. On the top, these are shown in magenta. The user can also perform a tissue overlay or a pairwise relationship overlay. The user can also use the portal to study drug-target interactions and explore potential therapeutics. Out of all human proteins, there are the proteins that we have annotated within Reactome. There are dark proteins, and most dark proteins are unannotated but can be viewed within Reactome. There are some dark proteins that have been annotated in Reactome, and I will walk through several case studies and show how the Reactome applications can be used to study both dark proteins, unannotated proteins, and compare them to proteins that have been annotated within Reactome. An example of a dark protein that has been annotated within Reactome is UNCLE. UNCLE is found in the adaptive immune system pathway on the Reactome site. The complexes or entities that are outlined in magenta are predicted to functionally interact with UNCLE. For example, the E3 ligases in proteasomal degradation list UNCLE here under the Molecules tab. On the Reactome IDG homepage, the user can view UNCLE in a functional interaction view with other pathways that are predicted to functionally interact. There is also a table view that lists information about the pathways that are predicted to functionally interact, such as the pathway ID in Reactome and the common pathway name. Most dark proteins have not been annotated within Reactome. The user can study unannotated dark proteins by seeing how they interact with the Reactome proteins that have been well studied and annotated. An example of a dark protein that has not been annotated within Reactome is TANK1, and there are multiple applications for the user to see this protein within the context of well-known annotated Reactome pathways. On the Reactome IDG homepage, the user can use the interacting pathway plot to see how different pathways are predicted to functionally interact with TANK1. Top-level pathways are shown on the right and are color-coded so that their sub-level pathways correspond on the plot. The user can click on sub-level pathways and open them here on the Reactome IDG pathway browser. Again, the entities and complexes that are predicted to functionally interact with TANK1 are highlighted in magenta. The user can view the different molecules of these entities, and the target development level shows the color coordination for these molecules. For example, dark proteins are shown in red, and the proteins that are seen to functionally interact are shown in magenta. Here is an example of our overlay data tool. The user can use the tool to overlay tissue-specific information from a variety of different tissues. They can select up to 12 tissues to overlay at one time. The user can also overlay pairwise specific relationships from different data sources, such as GTEx or TCGA. In addition to dark proteins, there are also some proteins that are just understudied or unannotated within Reactome, and the user can still study these. An example of a protein that is unannotated in Reactome is LARC15. After searching for LARC15, the extracellular matrix organization pathway is one that is suggested to functionally interact with LARC15. Here, LARC15 says it has one interacting protein flag, and this is MMP14. LARC15 is a fibroblastic marker, and MMP14 is known to be involved with extracellular matrix components. The user can also explore drug targets by overlaying the drug targets here. Drugs are shown as purple hexagons and can be overlaid on the functional interaction map. In addition to the functional interaction view, a user can see how drugs are interacting with certain proteins in this diagram view. This is beneficial to the user because a drug may affect a protein more upstream or even have downstream effects. Between the ReactomeIDG and Reactome website, a user has multiple tools to explore dark and annotated proteins and their functions. A user can explore a protein's function by seeing how it functionally interacts with well-known pathways. A user can perform a tissue or pairwise specific overlay and study drug targets and potential therapeutics. If you have any other questions about the ReactomeIDG project, you can find me at the poster presentation, and please be sure to visit idg.reactome.org and reactome.org. Thank you.